Okay. Right, okay, so we just need to sort out anaerobic respiration, and it's a lot trickier than you would think, really. Because what we know about anaerobic respiration is it's respiration without oxygen. So that's the odd thing about this. Is if we look where oxygen is actually involved, it's only involved in one place. I'll just reinstall it. So it's here, the half O2 is there. And so what we say is, well, if we take this away, okay, I can get that, that's not going to happen. Okay, that's fine. So this step doesn't happen. And that's a logical and sensible thing to say. But what we do, or you may recall from GCSE, is that with anaerobic respiration, we have, okay, so respiration without oxygen, 1, produces lactic acid, 2, we'll have to sort that out. It also produces a very small fraction of the energy. I don't think we do it at GCSE anymore, but we used to say it produces 1/18th, 1/19th, 1/20th of the energy that you get from aerobic respiration. And so the odd thing about that is we're saying, okay, so what you're saying is without oxygen, it has a catastrophic effect on the yield of ATP, but oxygen's only involved there. Okay, so that just needs a little bit of explanation as to why that happens. And the end point we're going to here is that without oxygen, only glycolysis can work. Okay, so we just need to explain why is it that without oxygen, only glycolysis can work. So the first thing, let's just deal with what's going on here. There's a step-by-step thing going on here. So no oxygen means that these electrons, remember oxygen, very key point, remember, it's the final electron acceptor. When any reference to anaerobic respiration, explain what's going on, almost certainly will involve, oh, no oxygen. Oxygen is the final electron acceptor. So, no final electron acceptor means the electrons can't be passed on to the oxygen. Can you see okay? Yeah. Consequently, this hydrogen pump, or this protein, has those 2 electrons and can't get rid of them. Which means that this electron carrier can't pass on its electrons to the final protein, hydrogen pump, proton pump. Wrong, there was not hydrogen pump, proton pump, or hydrogen ion pump. Which means this proton pump can't pass on its electrons, this one can't pass on its electrons, and the same with this one here. So what that means is, the reduced NAD can't give up its electrons to this proton pump here. As a consequence, this reaction can't happen. So we have an issue. So this can't take place. Which means we get a build-up of reduced NAD because it can't give up its electrons. So if you like, there's a queue of reduced NADs waiting. Come on, we need one of these to become available to lose its electrons in order for it to be able to give up its or pass on its electrons. If we get a queue of this generated, it means that we then have a lack of this, of NAD. It's NAD in its oxidized form. What we say is, because of a lack of oxygen, the electron transport chain can't function, and therefore we are not able to re-oxidize NAD, to get NAD back to its oxidized form, so we don't have any of that. As a consequence, that can't happen, and that can't happen, and that can't happen. Same applies for FAD, okay? So that can't happen, that can't happen. All of these stop working because we have no oxygen. And we would then say, well, hang on a minute, on that basis, this can't happen. Because you just said that lack of NAD means that this can't take place. And yes, that would be normally what would happen. But there is an alternative pathway. And the alternative pathway, so to clarify, electron transport chain can't work, Krebs cycle can't work, link reaction can't work, glycolysis can. And the reason glycolysis can is because we have this alternative pathway in place. I'll just change the way I've shown these. I've done that wrong. Just the way I orientate these. So makes this easier to show. So, the alternative pathway, we're going to result in the production of lactic acid. And what does that involve? That involves reduced NAD. Rather than being involved on the electron transport chain, the reduced NAD will give up its hydrogen, therefore forming the reoxidized version, N of NAD. And by giving up its hydrogen to the pyruvate, it forms lactic acid. So if you look at the structure of pyruvate, and I think pyruvate is that's pyruvate. And lactic acid is the same molecule. Let's start with that. It's an acid. There's our acid group for chemists. We did have that, but now we add 2 hydrogens on. That's lactic acid. So the 2 hydrogens that were with the reduced NAD will be passed on to pyruvate, and pyruvate then, in doing so, that's pyruvate, that's now lactic acid. But that's it. What it means is we can still produce those. Well, and of course it can happen on both sides, so we can still produce those. We lose 2 ATP still, so we still have, just glycolysis can take place. Consequently, as a result of anaerobic respiration, we generate, I hesitate to put them anywhere else, so as a result of anaerobic, we generate 2 ATPs. That's it, that's all we get. 2 out of a possible 38. That's 1/19 of our possible ATP, because anaerobic respiration can, because when things are anaerobic, glycolysis can still work. But there's a sorry, but there's a nice sort of order of events that take place here. So. As well video. So look, here's the order of events that we got to, I suppose it's a kind of, I say learn because it's not learn because you can understand it, but little waypoints that you've got to hit when we're talking about anaerobic respiration. Okay, so common question, words to the effect of explain what happens during anaerobic respiration, or explain why we only get a small yield of ATP during anaerobic respiration. Okay, so the waypoints you've got to hit here. So first of all, there's no oxygen, just a reference point for I know what anaerobic is, and it's the final electron acceptor. It means the electron transport chain can't function. Okay, that's it. We don't have to go, well, it can't pass on its electrons, no, electron transport chain can't function. That's fine. It means that NAD can't be reoxidized, or reduced NAD might be better there. No, it's okay, it's okay. The NAD can't be put into its oxidized form. That means the Krebs and the Link reaction can't function because we need NAD all the way through it. Only glycolysis can work, and this is why. Pyruvate accepts hydrogens from reduced NAD, forms lactic acid, and we only produce 2 ATPs. And then there's I think there's one more point. Oh yeah, somewhat separate. It means we get an incomplete breakdown of glucose. And there's still energy in those bonds. I mean, that ultimately, more familiar to chemists probably, and probably you'll have a better knowledge than me, but the whole idea of getting energy out of these molecules is there is energy in these bonds. Yeah? It's like, is it bond energy? Or is that a separate thing? There's energy in the bonds, and that's what the energy is. It's in the bonds within the molecule. Am I right on that, chemist? Yeah? So, if you think of what we start with glucose, okay, so think of the structure of glucose, loads of bonds in there, that's where we're getting that energy from. By the time we've finished the whole process of respiration, the glucose has gone. All those carbons have been released, and so the glucose molecules effectively disappeared, and we get 38 ATPs. Glucose hasn't disappeared yet. We've got 2 of these. So in actual fact, if you remember from GCSE, we go into oxygen debt with anaerobic respiration. When oxygen comes back and we stop doing that vigorous exercise and we come back to aerobic conditions, this can go back the other way. reform the pyruvate, and pyruvate can just go back to entering the Lincoln-Krebs cycle again. So if you think about how it's going to work, so I'll put this back on. If we then reintroduce oxygen, yeah, this can function again. We can release. This can now function normally. We can get NAD in its oxidized form, and what will happen then is it will encourage, it's the same thing, reversible reaction. It will encourage this movement. The NAD will actually do that instead. I should probably link that down to here, but okay. We can do that instead. Reform the reduced NAD, which can then go on to do that again. We can just get back to that situation we had before where it was working nicely, I suppose. But by reintroducing that, we're paying off the oxygen debt. And all that energy in lactic acid can be used again. So we sort of think lactic acid is wasted. It's not. You can get the energy from it. Just that idea of an incomplete breakdown of glucose is not uncommon to see on mark schemes. Okay, it's not uncommon to see it. One final add-on, that's animals. Okay, that's animals. It is different, subtly different, in plants. Any questions on that first before I... Okay. Just a slight difference, okay? Plants and yeast. What's the product of anaerobic respiration? If I say yeast, ethanol, okay? So we've got to get to ethanol. There's just a different step in there. So the first thing that happens to the pyruvate is it gets decarboxylated and it produces a molecule of ethanal. And then ethanal is converted to ethanol. And that happens in exactly the same way. Ethanal accepts hydrogens from reduced NAD and it forms ethanol. So the other class of that, so ethanol I know is again. So the non-chemists here, don't worry. It's just I just think it helps when you understand it. And ethanol is an aldehyde, yeah. Which is it that aldehyde? Yeah.
It does help when you draw the line. Like it puts it in perspective.
Well, when it works, because it doesn't always, absolutely, I couldn't agree more. Particularly if you know your chemistry and you can see, oh, that's an aldehyde, oh, that's an alcohol, and you can see how, oh yeah, that makes sense. It just picks up the hydrogen. Yes, I agree. It doesn't always work, but agreed. So you can see we just gained 2 hydrogens. Yeah, so this, in fact, we're adding a hydrogen on there and another one there, and we end up with that molecule. I'll take that off. It'll be that. Yeah. So you've just got to be careful that the context is set. To talk about, it does happen. I've seen it before, asking about describe anaerobic respiration in yeast or plants, whatever it was, and you just default, unfortunately, to lactic, to the one I had on first. You forget that no, this is just subtly different. Okay, that's it. That's anaerobic respiration. We've only got one more thing to cover, which is respiration in fats and proteins. Okay, or respiration of fats and proteins. How do they fit into the pathway? But I don't expect to see those on your diagram. So that's really I'd like that included in your diagrams. Any questions on it?
No, I have a question on
yeah, okay, I'll stop this.